Hello, everybody. Thanks for continuing to join me with the ATI TEAS 6 study course. This is the second installment of the science portion of this program. So the last lecture went by really fast, a lot faster than the previous one. I'm super pleased about it. So without further ado, let me share my screen and we'll get going. So, life and physical sciences. This portion of the exam is 8 questions. So these questions can vary from anything with describe basic macromolecules in a biological system, compare and contrast chromosomes, genes, and DNA, explain Mendel's law of heredity, recognize basic atomic structure, explain characteristic properties of substances, compare and contrast changes in states of matter, and describe chemical reactions. So let's move on. Describe basic macromolecules in a biological system. Just like before, pause if you need to, familiarize yourself with these key terms. If anything doesn't click or anything's not looking quite right, reach out to some of the other study tools that I've given in previous lectures. But we need to have the knowledge of carbs, lipids, proteins, and how macromolecules function in a biological system. Again, with this, I literally took the picture out of the book. For me to try to copy all of this would have been really difficult. So if you want to pause this and do a screenshot or something and fill this table out later, you can, because I want to focus on questions 1, 3, and again, so 2 and 4 you can do on your own, but I really want to focus on questions 1 and 3. Which of the following is the best description of the characteristics of an enzyme? Enzymes reduce the initial energy required for reaction to take place. The enzyme complement is similar in different cells of the same organism. An enzyme can operate under a variety of conditions to carry out a reaction. Enzymes are composed of monomeric units called monosaccharides. And again, I'm just going to give you the answer and we're going to walk through it. The correct answer is A. Enzymes reduce the initial energy required for a reaction to take place. So enzymes are a catalyst within the biological system. They just help speed up the process without basically using any additional energy for the reaction to continue. So it's kind of like, well, if you're trying to start a fire and you have your flint and your stone and you've got your kindling, you're trying to start your fire, Well, if you put lighter fluid on that kindling, it's going to light faster. The lighter fluid is kind of like enzymes in our body. Enzyme complement is similar in different cells of the same organism. That is not true. That is not accurate. Enzymes are transcribed and translated from genes, but the complement of enzymes in different cells is different, and it's based on the specific cell's function. So they can't operate a similar way in different cells of the same organism. They're basically assigned to what they're supposed to do. Enzymes can operate under a variety of conditions to carry out a reaction. Enzymes are highly reactant and product specific. So again, they can only operate in the condition that they're assigned to. They work under a really narrow set of conditions, and it depends on the enzyme. So a specific enzyme can do a couple of different things, but it's only what they're assigned to do. Enzymes are proteins and are composed of monomers called amino acids. They're not monosaccharides. So again, if you wanted to fill out this table to help you understand the properties of macromolecules, I highly advise it, but we're going to move on to question 3. Which of the following is a carbohydrate with structural functions? Gluten, lipoprotein, glycogen, or chitin? And the answer is D. Chitin is a nitrogen-containing carbohydrate that's an important constituent of cell walls of fungi and animal exoskeletons. Gluten's a protein, Lipoprotein is made of lipids and proteins, hence the name lipoprotein. And glycogen is the storage form of carbohydrates in animals. So again, if you want to pause this and define the following terms associated with enzymes, I highly advise it so you understand their function and what they do. Compare and contrast chromosomes, genes, and DNA. Okay, so pause, familiarize yourself with these key terms, because to understand these questions that you may encounter with the TEAS, you need to understand the function of chromosomes, genes, DNA, explain the relationship between chromosomes and genes, and differentiate among the structures of chromosome, genes, and DNA. So again, I took this literally out of the book. In which of the following directions is DNA coded? Left to right, top to bottom, 5 to 3, or 3 to 5? And option C is correct. DNA is read from 5 to 3. There's no left and right or top to bottom in chromosomes. It's the position of the chromosomes and how they're coded. So it's 5 to 3. Question number 2. A segment of DNA that contains a word made of 3 nucleotides is referred to as which of the following? A gene, a codon, a chromosome, or a genome? And option B is correct. A codon is a 3 nucleotide word that codes for a specific amino acid. A gene is an instructional sentence made of words. A chromosome is composed of a length of DNA incorporating a linear arrangement of genes on each strand. And a genome represents the sum total of all the chromosomal information in the cell. So I'll explain that more as we move on to the next sentence, or the next question. We need to match the letters shown in the figures to their description. The nucleotide is the letter, the codon is the word, the gene is the sentence, the chromosome is the chapter, and the genome is the book. So it's kind of like that feather on the bird and the egg and the nest and the nest and the limb, that whole thing. And the green grass grows all around. So we're looking for the letter in the word, in the sentence, in the chapter, in the book. So if we're looking from smallest to biggest, D is our nucleotide. D is our letter. E right here is our codon. This is our word. So this is our letter within the codon. Within the gene right here, the sentence right here, the double helix. In the double helix, the sentence is in the B chromosome, the chapter, in the book. So we have the nucleotide in the codon, in the gene, in the chromosome, in the genome. Hope that makes sense, how those all kind of trickle down and fall. Again, if you need additional support, Khan Academy has really good lectures that help understand chromosomes, genes, and DNA. Explain Mendel's law of heredity. Again, pause the slide, pause the video, familiarize yourself with these terms, and we'll move on because we need to describe the difference between dominant and recessive trait. Explain how gene pairs are inherited from parents. Use a Punnett square to predict traits of offspring, and describe the difference between inheritable and non-inheritable traits. The F2 phenotype of a dihybrid cross show a repeated outcome of 9-3-4 dominant to recessive phenotypes across several experiments. The ratio should be considered which of the following? Excuse me. Option D is correct. Non-Mendelian ratios being observed. The distorted 9-3-3-1 ratio with the last two numbers being combined to 1 is a non-Mendelian ratio due to interaction or other interference between genes. An expected monohybrid ratio would be 3:1. Expected dihybrid ratio is 9-3-3-1. A non-inheritable trait would not necessarily have a ratio that was repeated. So the repeated outcome of 934 is a non-Mendelian ratio that's being observed. A monohybrid cross of a plant with an unknown genotype is carried out with a homozygous recessive plant. The offspring show a one-to-one distribution of dominant recessive phenotype. The unknown plant was which of the following? Homozygous recessive, heterozygous, homozygous dominant, showing non-Mendelian inheritance. And option B is correct. The heterozygous plant with one dominant and one recessive allele would produce gametes with either one dominant or one recessive allele. Crossed to a homozygous recessive plant whose gametes are all recessive, this would yield a 50% dominant phenotype and 50% recessive phenotype. A homozygous recessive plant crossed to a homozygous recessive plant would yield 100% recessive phenotypes. A homozygous dominant plant would have two dominant alleles and produce all dominant allele containing gametes. When crossed with a homozygous recessive plant producing all recessive gametes, they'd produce heterozygous offspring with a dominant phenotype. And a monohybrid 3-to-1 ratio is only applicable to the F2 offspring of a heterozygous self-cross, so that has nothing to do with this. Moving on. Recognize basic atomic structure. So again, pause, review, and we'll move on, because you need to be able to label parts of an atom and understand the basic structure of an atom, describe an ion. Using a periodic table, identify the number of protons and electrons. So again, we have another table that I took literally directly out of the ATI TEAS study manual. Fill this out so you can understand it and understand how these elements correlate with each other. So again, I highly advise that you familiarize yourself, and not just with these specifically. This is just kind of to give you an understanding of proton numbers, neutrons, electrons, the atomic number, and the atomic mass. Which period in the periodic table represents which of the following? Shared atomic properties, increasing energy levels, decreasing valence electrons, and shared neutron numbers. Option B is correct. Periods show increasing energy levels. The farther an electron is from the nucleus, the higher its energy. Shared atomic properties are functions of groups, not periods. Valences are shared in atoms lined up in vertical columns. Periods represent increasing energy levels of atomic shells. The periods represent increasing electron numbers, not shared neutron numbers. I hope that makes sense. So moving on. A chemical bond between two atoms contains which of the following? One electron, two electrons, fused atomic nuclei, electron and proton fusion. So for a chemical bond to happen, there has to be two electrons. So we already know that A is incorrect. The correct answer is B. There needs to be two electrons shared to create the bond. Atomic nuclei stay distinct, so they're never going to fuse together. And electrons do not fuse with protons in chemical bonds. They just don't. Let's match these terms really quickly with the definition. And we can do that by breaking down the word and understanding the prefixes and the suffixes of it. So cation, anion, electron, neutron, and proton. So if we look at these words, you know, cation, anion, electron, neutron, and proton. So some things that stick out to me, like a negatively charged atom. So we're looking for something that is negative. And when I think of something, I think of like an antagonist. It's negative, like they're the bad guy, an antagonist. So a negatively charged atom is, excuse me, is, oh my gosh, is the anion. A negatively charged atom is anion. A positively charged subatomic particle. So something that's positive, it's pro something, it's for something, it's a proton. Proton is B, a positively charged subatomic particle. A neutral subatomic particle, again, neutron, is C. Electron is a negatively charged subatomic particle, and that leaves positively charged atom with cation, so it's positive cation. We'll learn more about the prefixes and suffixes when we talk about the English language arts portion, but this definitely applies here. So, explain characteristic properties of substances. Again, pause, familiarize yourself. If anything's sticking out that you don't understand, there's plenty of things out there to help kind of familiarize yourself with these terms. But you need to demonstrate the knowledge of density, demonstrate the knowledge of properties of water, for example, solubility, cohesion, and adhesion. Identify a substance using characteristics from a chart of given properties. And compare and contrast osmosis and diffusion. So, which of the following is the density of a substance that has a mass of 2.65 grams and a volume of 3.4 centimeters cubed? So the correct ratio of mass to volume is the weight divided by the volume, because we want to know how much of that weight can fit into the volume. We don't need to know how much of the volume can fit into the weight. So to figure this out, you have to divide 2.65 by 3.4. And if I can find my handy dandy calculator, 2.65 divided by 3.4 equals 7.794, blah, 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 blah. So we're going to just round up. C is the correct answer, 7.8 grams over centimeters cubed. That's the density of the substance. So we have to see how much mass can fit into the volume. Like, so if we tried to see how much water can fit into my water bottle, we need to know not how much my water bottle can hold. Because if we just try to, see how much my water bottle can hold and then just dump a bunch of water in, it won't work that way. We need to see the amount that it can hold first. So if we change the 26.5 grams to liters, like how many liters can this hold, or how many milliliters or ounces even into the circumference, not the circumference, but into the actual like area of my water bottle. Does that make sense? Because if I try to say, because my water bottle is this size, it can hold this much. We need to know how much it can hold in the size. I hope that makes sense. Which of the following is true about osmosis? Osmosis is the movement of water across a membrane. Osmosis is movement from a low concentration to a high concentration. Osmosis requires a large amount of energy to move water. Osmosis pertains to all molecules moving across a membrane. Oh my gosh, excuse me, I'm so sorry. Okay. The correct answer is A. Osmosis is the movement of water across a membrane. Like diffusion, osmosis does move from, osmosis is movement from a high concentration to a low concentration. So osmosis isn't going to move from where there's low amounts to high amounts. It's going to be the opposite. Osmosis does not use energy at all. It's just fluid moving across the membrane. And osmosis is specific to water. So all molecules cannot just move across the membrane with osmosis. Okay. Compare and contrast changes in states of matter. Again, pause, familiarize yourself, because we need to describe the states of matter. Demonstrate knowledge of the movement of molecules in the states of matter. Discriminate among different phases of matter. And I don't like the word discriminating. It's differentiating. I don't know, whatever. Explaining changes between states of matter. So melting, freezing, evap, condensation, things like that. Which of the following phase changes requires the addition of heat? Freezing, condensation, melting, deposition. And I hope that this kind of like, whoop, the answer just pops into your head, because freezing is a phase change from liquid to solid that requires removal of heat. You know, you can't put water outside when it's hot outside and hope that it freezes. Condensation is a phase change from gas to liquid that, again, requires the removal of heat. Deposition is a phase change from gas to a solid that requires the subtraction of heat. So the only thing that we can deduct from this with adding heat is C, melting. Which of the following is true of gases? Gases have definite shape and volume. Gases have no definite shape, but they have definite volume. Gases have a definite shape, but no definite volume. Gases have no definite shape nor definite volume. So the correct answer is D. Gases have no definite shape nor definite volume. They can go and move and do whatever they want. They do not have to maintain their shape, and they do not have a definite volume. The items that have a solid has definite shape and volume, because like this remote for my monitor here, it has a definite shape and it has a definite volume. It's not just like putty where I can move it and ply it and change it however I want it to. It has a definite shape, it has a definite volume. Liquids have no definite shape, but they have definite volume. So if you think about like my water bottle, like let's come back to this. Like let's say this is completely full of water. It has a definite volume. There's a certain amount of water in here, but I can pour it on my floor. I'm not going to, because that would be miserable, but I can pour it on my floor and that water can go wherever it wants to go. So it does not have a definite shape, but it has a definite volume. So if I have like 5 cups of water in here, or if I have like 4 cups, whatever, of water in here, and I dump it onto my carpet now has 4 cups of water. It just doesn't have the same shape that it did in my water bottle. There's no state of matter that has a definite shape and no definite volume. So C is just kind of irrelevant. Describe chemical reactions. This is the last slide for the science portion. So these are the last 2 slides. So key terms: acid, base, catalyst. Familiarize yourself. Understand chemical reactions, because we need to be able to identify covalent and ionic bonds. Simple chemical reactions. We have to describe how conditions affect chemical reactions: pressure, concentration, temp. And describe how catalysts affect chemical reactions. Again, that's kind of coming back to that enzyme thing. And know basic molecules. Example, H2O is water, NaCl, salt, CO2 is carbon dioxide, O2 is oxygen, C6H12O6 is simple sugar, and KCl is potassium chloride. Oh, I'm sorry. And we need to describe acids and bases in terms of pH balance. Sorry, forgive me. Which of the following substances contains an ionic bond? So an ionic bond, let me go back. Let me talk about it. We're going to move on. I'm just going to tell you the answer. Option D is correct. There is a metal and there's a non-metal. All these other options, both of these elements are non-metal, which makes them covalent bonds. Ionic bond is a metal and a non-metal. Question number 2, which is the following, in the following equation, what are the reactants? So if we can think about this little arrow right here, that's kind of like an equal sign. So we're looking for reactants and products. So if we can think of the previous side of the arrow, that's going to be our reactant. This is going to be what we get out of it. This is our product. So our reactants are AgNO3 NaCl. because option A includes all of the elements in the equation. Option C has one reactant and one product. And option D includes both the products. So we need to, the reactants are going to be on this side of the equation. So understanding that with chemical reactions will be really helpful. And then true or false, a substance with a pH of 3.7 is considered a base. And that is false. All acids have a pH that's less than 7. So 7 or below is considered an acid, and bases that have a pH greater than 7 is a base. So this would be true if this was, 13.7, but it's not. It's 3.7. So that's going to be considered an acid, not a base. Yeah. Let's keep going. Hold on, I've got 25 minutes on the clock. Let's keep going. Let's move through this. Let's just like power through, see if we can get it. Okay, now scientific reasoning. 7 questions. You have to identify the scientific measurements for using laboratory tools, critique scientific explanation using logic and evidence, explain relationships among events, objects, and processes, analyze the design of a scientific investigation. So, identify basic scientific measurements using laboratory tools. These are just basic lab tools and the measurements that we would take using them. So this objective includes identifying the unit of measurement in a model, diagram, illustration, photograph. Identify the numerical value of a measurement of an object. Selecting the tool necessary to measure volume, mass, or length of an object. Choosing a scale unit appropriate for the object being measured. So which of the following is the most appropriate unit to measure the distance length between Los Angeles and San Francisco? A liter, a meter, a kilometer, or a centimeter? Again, if we're going back to, excuse me, I'm so sorry. If we're going back to the King Henry died by drinking chocolate milk, K is kilometer. That is the highest we can go with the majority of the measurements we're going to see in the metric system. So liter is incorrect because that's actually measuring liquid volume. Meter is too small. Meter is not correct. It's too small to try to measure. I mean, I can measure a meter of a desk. How many meters is a desk? But kilometer is the answer we're looking for. Centimeter is, again, on that other end of the spectrum. That's the chocolate and drinking chocolate milk. So that's going to be one of those really tiny things. Like centimeter would be like, how long is your fingernail? How long is your pencil? How long, I mean, things like that. Like how wide is your cell phone? Things like that. That's going to be centimeters. Meter is going to be something a little bit bigger, and kilometer is the distance between LA and San Francisco. You're going to want to measure that in kilometers. So what does the prefix milli mean? 1,000, 100, 0.01, 0.001. So if we're looking at milli, again, millimeter, that's the milk. He died by drinking chocolate milk of King Henry died, that whole acronym. The prefix milli means D. Option A would be a kilo. Option B is hecta. Option C is centi. So D is the milli. So if we're thinking about a meter, 1 meter is 0.001 millimeters. No, I'm sorry, excuse me. My bad. I'm brain farting. It's been a really long day. The prefix milli means 0.001. I'm not going to try to elaborate. I'm just going to give you the answers. All right, so number 3. Which of the following tools could be used to measure the mass of a sample? A triple beam balance, volumetric pipette, graduated cylinder, or a ruler? And option A is correct. I'm going to tell you a funny story about it. A triple beam balance basically is where you balance the 2 weights on the sides. And my chemistry teacher, Dr. Gino, He loved to hassle the medical assistants and the people in the doctor's offices when they would say, "Oh, can you step on the scale for me?" And he's like, "The what? Oh, you mean the balance?" Because it's not a scale, it's a balance. So that was something that always pops into my head when we're measuring a mass is a balance. It's not a scale, it's a balance. Volumetric pipette measures volume. Graduated cylinders measure volume, and rulers measure length. So anyway, thanks, Dr. Gino. Okay, critique a scientific explanation using logic and evidence. Again, familiarize yourself with these. Let's move on. Logical conclusion based on evidence provided. Stated cause in a scientific explanation and stated effect in a scientific explanation, and evaluate evidence that supports the scientific explanation. So again, I took this literally straight out of the book. A researcher collects data on subjects in an experiment over the course of 1 month. Every day at 11 a.m., every subject takes 3 pills, A, B, and C, to control blood pressure. The following data are collected. So there's 4 clients. They have their initial blood pressure taken and their final blood pressure taken. So this is what they started with, and this is what we're reading after a month of experiments. So which of the following conclusions can be drawn from the data? Pill A is effective in treating blood pressure. Blood pressure went down for all subjects. The pill regimen is effective for the subjects. 11 a.m. is the correct time to take pills to decrease blood pressure. Again, I'm just going to give you the answers, and I'm going to explain why. So option B is correct. Blood pressure went down for all subjects. We're looking at 135/95 went down to 130/90. 130/80, 115/70. 140/90 went down to 120/80. 145/95 went down to 120/75. So it's congruent that all of the blood pressure went down for all of these people. Pill A is effective in treating blood pressure. None of these clients were isolated to pill A. They were all took the 3 pills every day. So A is crap. We have no idea if pill A is effective in treating blood pressure by itself. Option C, the pill regimen is effective for the subjects. Although that could possibly be true, we don't know what else these patients were doing at the same time. So like some of these patients may have reduced sodium from their diet. Some of these patients may have increased their water intake. Some of these patients may have cut out soda. Some of these patients may have started walking, some low impact cardio, something, anything. We don't know. All we know is right here, this data, and right here. So we don't know if it's just the pill regimen that's effective for these patients. We don't know. And again, 11 a.m., if it's the correct time to take pills to decrease blood pressure, we would only know this if we actually tested a different time. So these patients were just told to take every day at 11 a.m. They take their 3 pills, and we're going to measure your blood pressure a month afterwards. So what type of bias is present if clients 1 and 2 were given instructions to not exercise, and clients 3 and 4 were given instructions to exercise? This is an intentional bias. The researchers set up different practices for different clients without accounting for the effect, the potential effect on the outcome. So it's an intentional bias. It's basically saying, okay, you and you, you're not going to exercise, but you and you're going to exercise. That's an intentional bias. We're going to see if these change the readings at all. Which of the following term is used to describe a specific experimental condition that is changed to measure its effect? Data, variable, control, or bias? And option C is correct. Option B is correct. The condition that is changed to measure its effect is the variable. That's specifically the independent variable, because the data is just the outcome. The control refers to conditions that are kept consistent throughout the experiment. And the bias refers to a tendency of the experiment to influence results, knowingly or unknowingly, with the effective state. So the term that is changed to measure the effect is the variable. Hope that makes sense. And I'm not burning you guys out. Okay, explain relationships among events, objects, and processes. Cause, effect, and sequencing. The cause is what makes something happen. The effect is the result of the cause. And the sequencing is how we put them all together with their relationship. Comparing the magnitude of events, objects, and processes, determining the cause relationship between events, and sequencing an event or process will help you understand, will help you with these questions on the exam. Which of the following units is most appropriate for measuring the weight of an adult? This is no different than the measuring the length of LA to San Francisco. We're just using it on a different medium. We're measuring the weight of an adult. Grams, kilograms, milligrams, or micrograms. Again, micrograms are even smaller than milligrams. So the K in King Henry died by the grams as the base. Drinking chocolate milk, milligrams. We're going to do B, kilograms. That's going to be the appropriate way to measure the weight of an adult, is in kilograms. Because milligrams, micrograms are way too small, and again, grams are way too small. So which of the following units is appropriate for measuring the circumference of the Earth? Again, this is no different than that LA and San Francisco question. It's A, kilometers, because if we tried to measure in meters, centimeters, or micrometers, we're going to have way too high of a number. Even in kilometers, we're going to have way too high of a number because, let's face it, it's the circumference of the Earth. But we'd have a drastically lower number in kilometers than we would in meters. Which of the following can lead to emphysema, which is a severe respiratory problem? A high salt diet, alcohol consumption, smoking cigarettes, or sleep deprivation? Well, this is kind of where you have to use your critical thinking. Like, okay, does your diet have anything to do with respiratory systems? Well, possibly, but the thing that's going to stick out the most is smoking. You're literally smoking your cigarettes and you're inhaling the chemicals into your lungs. That's going to cause emphysema. It's C, smoking cigarettes can lead to emphysema. So you have to kind of think about that and understand the cause of smoking cigarettes can lead to the effect of emphysema. Sleep deprivation has nothing to do with respiratory problems. Alcohol consumption can affect respiratory problems, I'm sure, but again, nowhere near the effect of smoking cigarettes. Analyze the design of a scientific investigation. So again, this is something that we've been talking about with these statistical data, control variable, dependent variables, hypothesis, independent variable. So look at these key terms, familiarize yourself, because you have to be able to identify a relevant hypothesis based on a given investigation. Determine the strengths of a scientific investigation, determining the weakness of a scientific investigation, identifying dependent, independent, and controlled variables, determine whether a hypothesis is supported by evidence within a case study. Again, I apologize for the photo. It didn't come out very well. I tried to even scan it. It didn't work, so I'm just going to read it. Scientists believe that noise can affect the concentration of mice. They design an experiment to test this hypothesis. The scientists construct a maze and then have mice complete the maze. They test 500 mice. Half of the mice are required to run the maze with the same loud music playing in the background. The scientists then compare the time the mice took to complete the maze in silence versus the loud music playing. It is found that mice complete the maze in half the time when it is quiet. Which of the following is the dependent variable? The loud music playing, the number of mice in the study, the maze construction, or the time it takes to complete the maze? So the dependent variable, what depends on the other aspects is D, the time it takes to complete the maze. The loud music is the independent variable. This is what's being done to the mice. The number of mice in the study and the maze construction are variables, but these are controlled in the experiment. We're looking for the dependent versus the independent variable. The dependent variable is the time it takes to complete the maze. The loud music playing is the independent variable. Which of the following statements is the hypothesis? Mice finish mazes faster when the environment is quiet. If there is loud noise, mice will complete the maze more slowly. Mazes are an effective way to test mice. Loud music makes mice finish mazes faster. I'll say that 5 times fast. Loud music makes mice finish mazes faster. That's a tongue twister, not a hypothesis. Okay, option B is correct. Option A would be a conclusion statement after the data is analyzed. And options C and D are not supported by the experimental data. The loud music did not make the mice finish faster, and we have no idea if mazes are an effective way to test mice. We just did it because it was what we had available to us. So option B is correct. If there's loud noise, mice will complete the maze more slowly. Okay, Mary and Ellen do a study for their science fair project. They measure height and stride length of several people on the tarmac in front of their school and come up with the following data. So they did a study on 3 people. One person's 5 foot, one person's 5'6", one person's 6 foot. As the height increased, so did the stride length. For this experiment, identify the following: the dependent variable, the independent variable, the controlled conditions, and the possible hypothesis. So right away, the independent variable is, the dependent variable, what depends on the other, is the stride length. So a 5-foot person, it'd be really difficult to find them with a 3-foot stride. I'm sure it's out there somewhere, some way, somehow, but that's not what this study identified. So the dependent variable is the stride length. The independent variable is the height of the people. And the controlled conditions was the tarmac, the weather, how far they walked. So those are things that are the controlled variables that you have to include. And a possible hypothesis is that height affects stride length. How tall you are will affect the length of your stride. So anyway, moving on. Well, that is the last of science. Whew, we're getting it done, guys. We're getting it done. Thank you so much for sticking this out with me. You guys are great. I'm, again, trying to get as many of these done while my husband and kids are out of town. This is the quietest my house has been. I cannot tell you. But just, again, if there's any of this portion that you struggled with, I cannot stress it enough that Khan Academy is a fantastic resource. Crash Course is a fantastic resource. I don't know, just they're great at breaking things down into simple forms and helping you kind of check yourself for comprehension. So I heavily advise, using those in your study tools and keeping that in your arsenal, and using them when you're a little shaky with some things. I think that would be really wise. So the last lecture that I'm going to record is the English Language Arts portion, and that will be coming up next in just a few moments. But for right now, I'm going to sign off, take a break, probably go empty my urinary bladder, and we'll come back. Again, thanks for joining me. See you guys again soon. Bye.